IVPT, which is the IVPT sensitivity and selectivity, and I will start with the sensitivity study. The first question is, when should the IVPT studies should be conducted?
I think it's currently in the pilot. So when you can get some early reads if you want to, if you're doing transfers.
It's a part of development, I believe. Initially when we develop the method, we have to know like how long the permission studies need to be performed, and also if you have at least 3 doses running during the development of the method, you would roughly have an idea about whether the method is sensitive in terms of discriminating the performance at different doses. And then you would also get an idea like how long would you need to perform the permission studies if you had to pick up one of these 3 or 4 doses that you're using for sensitivity studies.
I think an important point to make sure that there's a clear distinction on, when we talk about the sensitivity, we're not testing whether the skin is suitably sensitive to discriminate differences. The skin is actual skin. It's going to either discriminate a difference or it won't. If it doesn't discriminate a difference, then that's the real answer. The skin does not discriminate a difference. Maybe you can create a difference in an IVRT, but you know what? It won't matter on skin. That's real. So we're not challenging the performance of the skin here. What we're evaluating is the method parameters, the IVPT method parameters. And the main consideration here is that if you're thinking about getting drug, if I'm a drug developer and I want to get a drug into the skin, I have some choices to make in terms of what strength. Why are topical products at 1% strength, 2% strength, 5% strength? 0.05 or 0.005%. Well, it's because it doesn't help to put more drug in there necessarily, because you're not necessarily going to get more drug through the skin. So just adding a higher concentration into the formulation, or doubling the amount of formulation that you apply, that doesn't necessarily get more skin in, because the skin is a rate limiting barrier. What the implications are then for the way you design the IVPT method is that there will be a range in which, as you increase the dose, you can see an increase in permeation, largely for mechanistic reasons associated with the dose. But the specific concern for bioequivalence studies is, what if your dose is so high that you're really rate limited by the skin? And even if the test product had a higher bioavailability, if the test product was actually dosed at a clinically relevant, much thinner film, lower dose, that it would actually have a much higher bioavailability than the reference product. But because the conditions of your IVPT method are using such a high dose, because you say, well, then it's good for my analytical, but I'm using such a high dose that I can't really pick up any more drug coming through the skin, and I lose the ability to see that, then my IVPT method is not sensitive to those changes, and we might draw the wrong conclusion about bioequivalence, because it'll look like, well, all test and reference behave the same. They had the same extent, but in reality, the IVPT method was not able to pick up that. And that's really what we're trying to figure out here. When we're figuring out what our target dose is during method development, we want to be able to make sure that if higher bioavailability existed, and one way to modulate that is with changing the dose, and I talked about it in my talk, then you'd be able to pick it up. And that's really what I think sensitivity is doing in this entire validation process. It's looking at the method parameters.
So what I'm hearing is, even though it's one of the critical validation, IVPT validation parameters, it would be beneficial if you conduct the study, once you identify all preliminary IVPT parameter and, the target dose. At that point, you work on, discrimination of the profile using either the modulation of the dose amount or modulation of the a dose duration, which we'll be talking in the next slide. But that is the point to conduct the IVPT study before you move into the validation or the pivotal study. Probably that will be the saving a lot of resources. If you have something and you need to optimize during the development, it would be a good idea to conduct the IVPT sensitivity study as a part of method development. So my next question is, what can be considered as an appropriate minimum number of donors and replicates for each treatment? I know there was no any specific recommendation in the PSG of acyclovir cream, but earlier Dr. Rene talks about 4 or 6 donor as well. Similarly, we also heard from Mr. Santos about 4 donors. What is your in your routine practice? How many number of donors and replicates do you use for each treatment?
We try to use 3, and if hopefully we have enough of the formulation or drug. When you are synthesizing your own drug and you don't have a tremendous supply, it limits your ability to do lots of testing. But I think 3 donors is something. But if something goes wrong with one, then you're not really sure what's happening. So the 4 donors is definitely a safe number.
Yeah, we use 3 donors in our shop too, particularly with 2 or 3 different parameters you're studying for sensitivity in the development stage. We try to keep it 3, and if we really need 2, we'll go up to 4.
Yeah, it certainly depends on how critical the data needs to be. Early methods development and just generalized screening, we often start with 3 donors and 3 replicates. But as we move into validation, it could be no less than 4 donors with either 4 replicates or 6 replicates. Again, partly a function of how well the drug penetrates through the skin. Certainly when you get to the pilot and to the pivotal, the more donors and replicates you have within reason of cost, expense, and logistics, the higher quality the data are.
Great. So what I'm hearing, I think it would be a good idea to have at least 4 donors, because sometimes when we use there is no any magic number, but if you is it wrong if you use 3? I don't know. Yes or no, but it's always a good idea to use 4 donors, as Dr. Stinchcomb mentioned, that if something goes wrong with the 1 donor, it's a good idea to have 4. Sometimes when we receive the application, we see these sensitivity studies are conducting using only 1 or 2 donors, and then it leads to a concern about the reliability of submitted data and eventually the IVPT method. My next question is, which drug products would be utilized to conduct IVPT sensitivity study? Is it the test product or the reference products?
Well, it should be the reference. That's what we used, because that's the thing you're trying to match, and so you need to figure out all the variation associated with that. If you're getting lots there, that you're going to at least expect that with your test. So that's typically what we use.
Yeah, we use RLD as well, because that's what you're trying to match.
Yeah, RLD too.
Sorry. Here, just one quick comment, right? So in terms of the number of donors, right? So I think one aspect no one mentioned, depending on your compound, you're going to have much higher variability as well, right? So we have seen over the years more hydrophilic compounds, they tend to show much higher variability. So to have appropriate conclusions, you may, of course, need to increase the number of donors and replicates. So just one last point.
I might also say that I think it's particularly advantageous to have donors, so multiple donors over multiple replicates. Although it does depend upon the nature of your data. That let's say you had a budget, and within that budget you could do 36 diffusion cells per treatment. You could do 6 donors with 6 replicates and get 36 diffusion cells, or you could do 9 donors, 4 replicates. I think you'd typically be better off doing 9 donors, 4 replicates, in terms of the quality of information you get. Now, that is unless the data is really highly variable, even within a donor, then you may want to have more replicates per donor. But I think this is the kind of consideration you want to have about the way that you parse out your diffusion cells among donors and replicates too. And then, what I'm hearing, especially for sensitivity studies, is that something in the range of 3 donors, 3 replicates may be reasonable. While 4 donors, 4 replicates may be, ideal and preferred, and maybe particularly more valuable when we get into, the pilot study. But in the method development work, it seems like, the evidence from 3 donors, 3 replicates, as long as that's what you finally end up with, is something that I think I'm hearing most of the experts would be comfortable with making a determination based on that.
And to support what Sam was saying about the donors, every once in a while you find a really anomalous donor, very low permeation, very high permeation versus the other 5 or 6. The more donors you have, the less influence on the overall mean that anomalous donor has. If it were 1 of 3, it would have a significant impact on the bias of the mean. If it's 1 out of 6, less influence.
And that's a really good point. And it also ties into the IVPT sensitivity study. If you've got only 3 donors and 3 replicates, and 2 of those donors, you don't see a particular discrimination, but in one donor, in one of those 3 donors, that's really contributing to where you're seeing the discrimination, then that data may not be as compelling. That's where that might have that 4th donor that plays a much larger role in providing the compelling evidence for sensitivity.
Thank you for your input. To further expand in terms of the study design, let's discuss about the different approaches to demonstrate IVPT sensitivity for the drug product under investigation. So there are 3 potential approaches, which include the modulation of dose amount, modulation of dose duration, or the product strength. Now, for the modulation of dose amount, the current recommendation is to have 1 higher and 1 lower dose amount in comparison to the target dose. Now, if you are working with a particular drug product, how do you select the lower and higher dose amount with respect to selected target dose? And what we have seen in terms of the ANDA submissions is, the current recommendation is to have your target dose between 5 to 15 milligram per centimeter square. Now, it really depends on the drug product, but what we have seen is, when you pick the dose amount, which is too close, so I think earlier Mr. Lemon talked about, 5, 10, and 15. If you pick that one, and if you're lucky, maybe for that drug product, you see the good discrimination. But what we see majority of the time, that when you pick the dose amount which are too close, it is very difficult to have a discrimination in the profile. So in your practice, how do you select the amount of lower and higher dose amount? And I saw there are multiple questions on the incoming chat asking, if you go on a higher side, how higher you go? Do you go 60 milligram per centimeter square? Do you go 100 milligram centimeter square? So what are your thoughts on selecting the dose amount using this approach?
So I'll jump in there, but I don't think that there's a problem with going substantially higher. In fact, you might need to go substantially higher because just the nature of the dosage form, the nature of its metamorphosis and drug delivery, you may not really be delivering much more to the skin unless you've got substantially different conditions. And if you have to create substantially different conditions, like a substantially larger dose, not just 3-fold higher, maybe 5-fold higher, something substantially higher, I don't think that's a problem. I think the point is you're trying to create a condition where there clearly was a higher bioavailability, and you're dealing with a small number of donors and replicates, whether it's 3 donors or 4 donors, it's a small number of donors. And to be able to tease that out qualitatively among the variability, you need enough of an increase in bioavailability. So, if you had to go up from your 15 milligram or 10 milligram target dose to up to a 60 milligram dose in order to be able to see that, I don't think that's unreasonable. one of the questions here is the lower and higher dose amounts. I think it's ideal when you can show in the sensitivity study that as you modulate the dose lower and higher, the IVPT response is lower and higher. That's ideal. But I think in terms of what's the real concern here, there's not a particular concern on the lower end. The very fact that your IVPT profile went from lower levels to higher levels, you're able to detect it at lower levels. It's the higher level that we're really concerned about, that you may get the wrong answer on interpreting bioequivalence data. So I think this is something that I'd be interested in. Is this a place where IVPT sensitivity studies can be streamlined in terms of, what are your thoughts on the value of that lower dose, especially because sometimes that lower dose may put you into a place where it's really hard analytically to get the quantification.
Now, I've started thinking more about not doing just one lower and one higher, but doing a couple lowers and a couple highers. Because if you just did the 3, like you said, it may turn out that at 5 milligrams per square centimeter, there's insufficient samples with detectable drug in it. So now you have to go back and redo another one. Or if your top 2, your 10 and 15, show the same rate of delivery, there's no discrimination. Well, then you have to go back and do some other ones. And it's difficult to do for 3 and then realize that that's insufficient to show discrimination. Then you have to set up another study, do 3 more with the same skin until you find discrimination. But here's the real question I've always had is, what is sufficient discrimination to be acceptable to claim that there is discrimination between doses?
So probably we can touch base, we can stay with the study design in this slide, and the next slide I'll be talking about the data analysis. So do you mind if we can come back to that question on the next slide, Mr. Lemon? Thank you. So what I'm hearing from you, are you suggesting or are you recommending to have multiple lower dose amount and multiple higher dose amount in terms of designing the study?
It partly depends on the logistics and timing and cost. If you do 3 and it turns out you can't discriminate 2 out of the 3, you're going to have to go back and do some more work. Now, if you can afford to do more work, clients willing to pay for it, you have sufficient skin from the same donors, why not just do the extra work up front rather than having to repeat and repeat and repeat? Maybe John Len has some observations that could be shared similar.
Yeah, I mean, in a perfect world, you do a couple doses below, but we're starting to get in a situation where at some point we should probably talk about the volume of work and cost. And in a perfect world, we test all that stuff. It's just it's a lot of extra work, and with that comes some cost. I think the challenge for me also is sometimes when these formulations are older and they're suspension based, going up, you're just essentially adding more crystals to the surface, and so it gets a little funky. So it won't be linear, certainly. And at some point, what you start seeing, the data can show this, is you start seeing a plateau. And I think to Paul's point, there's a small point to where it starts to plateau. Is that sufficient to demonstrate sensitivity? Do you want us to design the study for these suspension-based formulations so that you go up until you see that plateau and then you push it, that threshold up in volume? Again, that's stuff you can test, certainly you can test, but that's just adding more work.
Okay, so what I'm hearing from the 2 panelists is importance of using the lower and higher dose amount. And I know Dr. Lenny mentioned about, what information we are getting if we are having a lower dose amount. Are we, because I think he was referring to the streamlined IVPT sensitivity study, and he was, seeking the feedback on, do we really need to have a lower dose amount? So any comments, on the importance of having a lower dose amount to demonstrate IVPT sensitivity? Because at the higher dose amount, we are talking about, saturating the skin, and we are not getting any higher permeation profile. So that is kind of a rationale for the higher dose amount. If we like, in terms of the lower dose amount, does it provide any critical information about the optimized or developed IVPT method?
I guess for me,
Using the lower dose amount would be easier to get than the higher dose amount, but then you have all the assay sensitivity issues as well. So if you have a very potent drug, you might have issues trying to get the lower end as well.
And that's a great point. I think even we have a lot of question in the chat box that especially if you pick your target dose being a 5 milligram per centimeter square, and researchers sometimes struggling. I mean, if we go with the earlier presentation, we were talking about twofold higher or lower on the selection of higher and lower dose amount, or even go for the threefold. So if your target dose is 10 milligram, your lower dose is somewhere in the 3 milligram per centimeter square area. And do you get enough API permeated in your receptor compartment? So there are some analytical challenges on conducting the lower dose amount, especially with the potent drug product. So I think this is something we can have a broad discussion about how we want to do it. But the current recommendation is to have one lower and one higher dose amount. Now, in the interest of time, I know we have a lot of comment on that one, but I'm going to move forward to the next modulation, which is the dose duration. Now, Dr. Rene already covered about on what scenario we can go for the dose duration where you don't see your Jmax if you have a 96-hour study or 72-hour study, and if you, especially with the ointment when there is no metamorphism and your Jmax is still going up. My question is, how do you design your IVPT study if you are going with the modulation of dose duration approach? In other words, how do you select the wipe off time? What would be the wiping off procedure? Consideration for the frequency of sampling. Do you suggest to have a rapid sampling after the wipe off? Does the target dose wipe off need to be clinical relevant? So if that drug product is a once a day application, does the target wipe off should be around 24 hours, or it really depends on the IVPT method? So with that, I'll open the floor for discussion.
it's probably a situation where you're going to have to run a very small study, maybe a single donor, and look at multiple wipe-off times. Ideally, you'd have the data to see if you're not getting that kinetic profile already. And then if you have to move into this situation, you probably will have to do a couple. And again, whether you want to try to link that to the clinical, I think ideally that'd be great. The patient probably is not going to leave this thing on more than 12 hours. That's probably a good starting point. And then do you go before that and after that? But I think some of that will just depend on the product and the kinetics that you see early.
Yeah, I would agree with that. A small first study is going to give you a lot more insight. Even if you don't touch the product, if you apply on real human skin, the product disappears in 4 hours or so. It doesn't stay like the way it stays on the in vitro skin studies. But you also should get some insight from the formulation itself, if it's a hydrophilic formulation versus hydrophobic formulation. So some of these early insights, a quick run, and based on how fast you're getting the Jmax, etc., and the wash drug wipe off can be determined based on that.
And there's two factors to consider. If you're using some sort of solvent, you may inadvertently redissolve the drug and drive a pulse dose or a burst dose during the surface wash. You may end up with a double peak appearance, which could be a problem. And the other factor is how efficient that surface wash is or removal to ensure that you don't have, that you've removed essentially 95% or greater of the product from the skin itself.
I think that does bring up a really good point in terms of how the wash is conducted. I think it's really tough with ointments because it's hard to dissolve petrolatum in things other than heptane or something like that, which you really don't want to use such strong solvents on the skin. So I'd be interested. I've seen people use methodologies where it's a cream, and they might use a quick alcohol wipe in the same way that you get an alcohol wipe before you get a COVID vaccination or something. A brief exposure of the surface of the skin to alcohol. I'd be interested. How concerned are you? And I'm not that concerned about it in ruining the interpretation of the study. If you can get a quick and good clean at that point. I've also seen protocols where people will use cotton swab, Q-tip type of thing, so get the bulk formulation off, maybe dry and wet, and maybe once with a little bit of very mild soapy water to help to just get a little bit of that applied dose off. And I think you're right, Paul, that one might expect that in that process that that manipulation that you might actually solubilize drug that's sort of crystallized, dried out of the formulation, and you might pick up a little blip. Is it okay if that's the same for the test and the reference product, but at least what you've been able to do now is to be able to produce a profile where you can see the absorption phase into the skin, and then as that drug permeates through and the kind of elimination curve of the drug from the skin, and compare that for the test and the reference product. Is that meaningful data? What are your thoughts among this panel about using those kinds of wipe-off procedures, understanding the bargain that we're making here in terms of being able to make a good assessment between the test and the reference product?
Well, if I can push the question back, I mean, you've obviously seen more than we have. It seems to me like this will add a lot more variation because you're adding another step, another wash-off thing. Anytime you add something new in there, you're introducing potentially new variation. Do you see that? So maybe you do see the blip, but do you see an increase in the variation in the CV?
I don't think so. At least not with the data sets that I've seen. I haven't actually even seen the blip, but we know from old studies with steroids that could happen, that you can in the washing off procedure actually resolubilize drug and see that. But in the type of procedures that we've looked at, where we were trying to address the question, if your IVPT profile and method development just keeps rising and rising, what should we do? Should we just say, well, both the test and the reference products under these conditions have the same behavior and they're well matched, and that's the conclusion, and okay, Jmax is your last time point, but it was a sizable study and they both performed the same, call it good? Or do we say, well, let's go ahead and do a wipe off and see if the absorption and the elimination phase into and from the skin are well matched for the test and reference product? Is that more meaningful? And when we've done those, I think it's not that the variability is increased, not that we're seeing the big blips, but rather that when you come to do that for an IVPT sensitivity study, you really have to choose the duration of those wipe-offs very thoughtfully relative to where Jmax is. Because the ideal situation is, do the entire flux profile. And if you have a product, in fact, that gives you, and that, well, my Jmax might be at 18 hours, then you want to have your sensitivity such that your target dose is maybe at 12 hours, and then you have a lower and a higher, because if all of your doses are past the Jmax, you won't be able to differentiate them. You want to be able to have one where the higher dose is still able to deliver more drugs. So that's more, I think, the lesson we've learned from, and I'm not sure how clear that came across, and it's a little bit technical, but I think those are more the issues that we've seen with the data. Otherwise, my impression from the data we've seen is that this is actually, modulating this as a dose wipe-off is actually quite an elegant study design for being able to get very nice IVPT profiles, and often to be able to compare products in a relatively shorter time frame. We've done this both with IVPT, like in vitro, and we've also done the same studies in clinical situations. And I've been quite impressed with the data that we've seen.
Thank you. I think we can move quickly now. We have certain topics to be covered. So The next one is the modulation of product strength. I can share like, what we have seen. So based on the data we looked so far, we are, I think that based on the data that we have seen, the most of the time the either approach dose amount or dose duration, work for most of the time. So far we haven't seen a data where, the product strength is a better option than the dose amount and dose duration. So based on your experience, did you come across a situation where the modulation of product strength is a more suitable approach to demonstrate IVPT sensitivity?
Yeah, this is another one where when it's a suspension-based formulation, yeah, I don't expect it, and we don't see it when you go up in concentration. So this is another one of the situations when you get into those partial suspension formulations. Is it okay to go lower, have two lower strengths, and then a higher? Do you need to show the data that, okay, if I go up in concentration to a certain point, are you saturating? And then how far do you go up?
It also becomes practically not so possible if you're using RLD, and you can go change the concentration. It's not, spiking is not same as incorporating the drug in the emulsion.
That's a good point, yeah. So let's move on to the next part, which is the data analysis. So my FT fellow colleagues, I would like to have your perspective towards the IVPT sensitivity studies. Would you please share your experience for data analysis in terms of the expectation to demonstrate adequate IVPT sensitivity using selected approach? And also, I would be interested to hear from the external stakeholders from our panel. How do you interpret the IVPT sensitivity data in terms of qualitative or quantitative data analysis? Let's begin with our stakeholder first.
So here, who do you want to answer? And
so we can start with our stakeholders first. That once you conduct the study, how do you interpret the data? How do you refer to the guidance? And at what point you call your method being a discriminatory? And then probably somebody from the FDA can provide their input on how to analyze those data.
I think you have. I think if you have your panel members kind of quiet, they're looking for some help here. So I think it's this follow-up thing. How, is it qualitative? What is qualitative versus quantitative? Do you want us to run statistics on it? We're not expecting linearity. This is a challenge for everyone.
Sam, would you like to make a comment on that one?
I agree. I think it is a challenge. And I think what this comes down to is, if any of you who are in industry or in CROs were in the shoes of the reviewers, I think you're all scientists. You're all looking at the data. You're all trying to understand, do these results support a certain conclusion? And we're dealing with small data sets, a few donors, a few replicates, with enough variability in there that I think to get quantitative statistical results where the reviewer doesn't have to make a judgment call and just can say, well, it met a statistical endpoint and they are differentiated, would be lovely. But I think that would require a much larger investment in the study than I'm sure is warranted. I think this is a kind of incremental risk mitigation strategy so that you understand the performance of the IVPT method in terms of a variety of dimensions, in terms of its discrimination ability, in terms of its sensitivity. And so I think when we say qualitative here, my perception has been that there's two lines of evidence we're looking at. One is you're intentionally working with a given formulation. You're changing something about the dosing, the duration, the dose amount, in order to be able to alter that bioavailability. Are you sort of seeing that in your IVPT? Ideally, I think you can, in most methods, develop it in such a way that when the flux curves are compared, that while maybe not the entire region across the flux curve, but that you'll have regions maybe near Jmax where the curves are differentiated beyond kind of their error bars, and you can say, okay, I'm seeing a discrimination here. and with a different bioavailability, I would pick that up in my pivotal study as being different. I'm reasonably compelled that we've got the method developed well. And then you complement that with data from the pilot study with that third formulation, that altered formulation, or that different formulation, which also shows itself to be differentiated. And that may be wildly different, maybe a completely different dosage form, but it's differentiated. And then you use both those together to say, well, I think collectively, if the test product had a different bioavailability, I think the IVPT would pick it up. And I have no reason to think that I wouldn't see it, because when there are differences, I'm seeing it under these different conditions, just looking at the curves. I think there probably are statistics that you could run, but or kind of other kind of quantitative analyses that you could do. And if some of you have suggestions for that you think would really impact, would really change your comfort level with that IVPT method, we'd really love to hear about that. If not, then what's your comfort level? When you look at the method and go, yeah, I think we've got a good method, what do you want to see about the sensitivity or selectivity of the method to say, yeah, I think we've got it? What compels you?
Well, one
And
problem.
go ahead.
Sorry, go ahead.
Please go ahead.
One problem I encountered was I thought there was sufficient discrimination between dose amounts, but the reviewer came back and said it was qualitatively insufficient to discriminate. So now it's my opinion, your opinion. When that's the difficulty I have with there's no standards of degree of difference, the minimum degree of difference needed to establish an appropriate trend.
So I think this is the problem that we're trying to resolve with this workshop. So this is a sophisticated issue. I'm not sure that I'm hearing that someone is a proponent for inflating the size of these studies to the point where we can make unambiguous calls on the data. So if we're going to make judgment calls, what are the principles that should go into the judgment calls? How what made you think the data was good? Did you feel like, look, the error bars are separated here, I think we're good? Did you have some kind of other way of looking at the data? I would love to hear from you. And I hear the silence, and in the course, if the silence continues, then the consequence will be these kinds of discrepancies in judgment. Because if you, as a panel of experts, don't know how to figure this out, then we'll be stuck with this ambiguity.
Yeah, so I certainly think it's gonna be not impossible, but with small data sets, getting a statistical analysis, which is more obvious, I think is be a challenge, and I'm not sure it's worth going through all that extra effort to get the sensitivity. Then it means you need to be more qualitative. And then what do those differences mean? Which I think you also need to feed back into what is that product again. Not to harp on this again, but there are some product-specific things that occur, especially with these suspensions, that you don't expect to see these differences. Therefore, that should factor into your rationale of what you consider qualitative. And I think if the people that are on this phone that are doing these studies all day, every day, are saying we think this has sufficient sensitivity, then it's going to be a conversation, I guess, between submission and reviewer.
So let me put this out there. If you've got 3 different dose amounts, as an example, and you've got 3 different curves, and the kind of line connecting the curves, 3 different colored lines are differentiated, but the error bars are completely overlapping, I'm not compelled by that data. Are you?
So, Dr. Rani, I think we can, keep discussing this one on the next session where we have a statistician in
That
the
sounds
panel.
good, yeah.